Hello, sorry, I'm having a bit of difficulty here getting my sound and sharing, but good morning, good afternoon, good evening. I hope everybody's doing well. It's a pleasure today to have Natalia, Dr. Natalia De Leon, who is a professor at the University of Wisconsin in Madison. And Natalia's research has focused on plant breeding and quantitative genetics, specifically maize improvement for productivity and resilience. And she explores the interface between plant breeding, quantitative, and molecular genetics, combining different sources of information like genotype, genotypic, phenotypic, and expression data. Also, Natalia conducts genetic analysis of development traits in maize. She has a master's and a PhD from the University of Wisconsin in plant breeding and genetics. And before I give you the floor, Natalia, I think I comment this to you. We already clarified before the opening of the seminar that you are Uruguayan, so we have the same nationality. But also, I wanted to mention that when I started working in plant breeding, I worked for 5 years with the Uruguayan maize germplasm collection that your dad did in the 70s, which to me is very close to my heart. So we do have a good connection there. Natalia, it's a pleasure to have you with us. I'm gonna stop sharing here and give you the floor. It's nice to have you with us.
Well, let me first make sure I have the slides and that you all can see it. Are you able to see my
Not
slides?
yet. Not yet.
Um, can you?
Now it's coming. Yes,
Okay,
now we can
perfect.
see it. Okay. Thank you.
Perfect. Thank you. Well, thank you so much for the wonderful introduction. And I have to say that you have met the best maize person in my family, which is not me. So it goes downhill from here. So with that introduction, I will say you get the second or probably third worst version of plant breeders in my family, which is a story for a different time. But thank you so much for the opportunity to be here. It's a great honor to be able to be here and talk a little bit about this work. And I have to start by saying that what I'm going to talk about today is the results of several conversations and experiences with so many collaborators that I probably will not will do it justice. I have pictures of certain people that contributed specific pieces of data that I will present. But primarily, I work very closely with my collaborator, Sean Kepler at the University of Wisconsin-Madison. And a lot of what I'm going to talk about is the result of endless conversations while pollinating corn in the field over summers and thinking about the nature of important, economically important traits and what is it that we do as plant breeders in the sense of thinking about where variation comes from and how we make decisions about how to harness that variation, in the present and then moving forward. So as the title of my presentation indicates, I'm going to pose this question of why a lot of the traits that we look to improve in plant breeding seem to be controlled by a large number of small effect genes, and whether that situation is the cause or is a consequence of how things have happened over time. So an absolute pleasure to be here. And let me see. Okay. So I don't want to preach to the choir, but one slide to put context of where we are and where we need to move towards. The challenge is real. I don't think I need to say that to this audience in terms of where we will be in just a few years from now in terms of the number of people that will be habitating the world, and then the real challenges that are facing the world, but especially those of us focused on improving food, feed, and fiber production with challenges of having less water, variable climate, and degrading soils. And as this relatively recent study has shown, the challenge in terms of what we need to do practically in order to meet those challenges and address them that, in order to produce the energy or the food that we need for that growing population will require a very large increase in crop calories in 2050, that the amount of land that will be required in order to make that happen, which is estimated to be twice as big as the whole country of India, and then the issues of the greenhouse gas emissions that are affecting the globe, and the need for addressing it in a very urgent manner in order to not even address it, but at least mitigate or not exacerbate the current problems. So what I tell my students when we are talking about these issues is that all of these is overwhelming just in and of itself. But if you think about the timeline that we have for these, it has taken about 10,000 years. And I'm here using the example of corn, and that's what I focus on, and to make it clear that it's the only cartoon that I know how to draw in PowerPoint. So you will see a lot of not very professionally looking corn plants. That is all on me. But it has taken about 10,000 years to domesticate a crop like maize from its ancestor Teosinte. And if you do the math, between now and 2015, we have about 10,000 days to not only continue to address the needs for more food and fiber and feed food and fiber for the world, but also do it in the context of all these other challenges that need to be addressed in order for us to truly be successful. So then what do we do? If we think about breeding and how that, what we do will connect to these issues, we think about where is the variability that we have available to work and improve all of these different traits. And we have a significant amount of variability that is theoretically available to us, that has been generated by recombination and segregation. And we are constantly in plant breeding, of course, in a constant exercise of allocating resources to identify and select what we consider to be the ideal genotype. So one of the things that I like to do in my introductory plant breeding class is to put things in perspective of this idea of what we call the breeding lottery, that again was described in this paper that I'm showing here on the top of the slide. If we're dealing with a trait that is controlled by one locus, and we are looking at an F2 progeny, and we are trying to identify that homozygous individual with those favorable alleles, we have 1/4 probability of identifying that ideal genotype. If the trait has 3 loci, all of a sudden we now are down to 1/64 probability of identifying that favorable ideal genotype. With 5 loci, it's 1 on 1,000. How many of you select one individual of populations that have 1,000? I think we are getting to where the majority of our breeding populations sort of exist. And, thinking about a trait that in my dictionary is not the most complex, which is, something that is controlled by 15 loci, it is actually more likely to win the jackpot. This is, one of those federal national gamble games here in the United States. And it is more likely to win the Powerball jackpot than to be able to identify that ideal genotype if we are dealing with a trait that has 15 loci. And we know that, for example, an average genome in the case of maize is estimated to have 30,000 genes. So it's not a minor task to think about, what is it that we are really trying to do when we are working in the context of breeding. So what is the type of variation that we have access today? And my argument is that a lot of the variation is historical in nature, whether we are thinking about the impact of domestication, again, through the process of 10,000 years of selecting for traits that have transformed those wild species into our economically important domesticated plants, or the more recent history of hundreds of years of breeding, modifying plants to meet the needs of our population. And so when we think about, what are the traits, what is the variability that is left after this entire process, this is a graph from a work done by Chuye Chen. She's a research scientist in our group, and this work is part of what she did when she was working with John Dobley and Jim Holland and Jeff Rossibera as part of her PhD. And the short version of this graph is that if we compare, we have matching populations, you can think of maize and teosinte. What she identifies that the QTLs that she looked at the genetic architecture of different domestication traits between maize and teosinte, and there is a much smaller number of QTL that have genetic variation for maize that have been selected during domestication. So a large number of those QTLs, and there is a very, very large number of QTLs controlling each one of these traits. So we see how the variation has been eroded. This is just one example to illustrate that during the process of domestication and breeding. You might argue, however, that we do every year have an enormous source of new mutations and recombinations if we think about the typical breeding populations. And again, I'm just here contrasting in any given year. So if you look at the Milky Way, it is estimated that there is at least 100 billion stars in the Milky Way. Well, there are 20 times more corn plants grown in the United States every year. Each one of those plants, of course, is the product of different effects of mutations and recombinations. So we might say that could be an endless source of variation. The reality is that the plants of maize that are actually used to contribute to the next generation are orders of magnitude a smaller number. The plants that we actually can pollinate and use to start new populations are substantially smaller. Then, just again, kind of thinking about my introductory plant breeding class, we might say, well, but what types of alleles? We know that neutral alleles remain in populations for long periods of time, and that is true. So this is a graph that I use in my own class, and these are just simulations when we start with alleles that are neutral in nature. In these examples, a population size of 1,000 individuals, and looking at a relatively long period of time with 500 generations. We start at an intermediate frequency. We are going to see that these alleles will indeed remain in those populations for relatively long periods of time. And I'm just growing random populations, 10 random populations in this particular example. Now, what happens when those alleles are not present at intermediate frequencies? In this example, starting with an individual mutation that has a small effect and has a relatively small frequency, the reality is that these alleles are very likely to be lost within a relatively short period of time. Estimated in here, between 10 and 20 generations, almost every allele will be lost. You can see 1 out of 10 of them survive and it remain at a very low frequency. The other side, or the flip side of that, is when the mutations have very large effect and we start at very small frequencies, they are going to sometimes be fixed. And I have to say, it took me pushing the button about 20 or 30 times in order to get these two populations to really rise to a fixation level. But we will, in some few cases, have fixation of these alleles, but the majority of the time they will still be lost. And those that, again, are fixed relatively quickly in these populations. So just kind of summarizing this introduction in what is the variation that is the substrate for breeding. Large positive mutations are likely to be rare in the context of breeding. If a mutation is beneficial in any given population, it is likely to be quickly fixed, and therefore it is improbable that new functional variation is excessively or currently available in large amounts in the typical breeding programs. So what we really have to work with is historical allelic variation that is the predominant substrate for our breeding efforts. So much of what we have in our hands for current breeding efforts are based on small effects that are there from historical reasons. And the other detail is that these effects are also be found to be very frequently linked to negative alleles. This is a paper that was published a few years ago now, but a really beautiful work done by Ed Buckler's program, a group looking at the nested association mapping population in maize. And what I'm showing you in here and the figure on the left is this nested association mapping population is constituted of 25 different recombinant imbrine lines that cross 25 very diverse parents with the reference parent B73. And what you're seeing here in the horizontal figure are the 10 chromosomes of maize. So those gray lines are separating the 10 chromosomes. And what you see in colors there are places where we have significant amount of segregation distortion. These being a recombinant imbrine line population between two parents, the expectation is that most of these loci would be at 50%, each alternative allele. And it turns out that we see a number of regions in these genomes that have this segregation distortion, showing that there is very uneven rates of recombination in these different populations across these different chromosomes and populations. And the other interesting finding from this work is that when this group look at what is the residual heterozygosity found in these populations, and then comparing the centrometic regions in these 25 biparental populations compared to the rest of the chromosome, it has found that across all 10 chromosomes, there is actually an enrichment for heterozygosity in those centrometic regions. Of course, this is sort of like a confirmation of the the old proposed theory, dominant theory of heterosis, with corn being a primarily hybrid crop in many parts of the world. What they found is that there is a large number of heterotic regions that are concentrated in these centromeric regions. And in those centromeric regions, these important alleles are actually in repulsion phase linkage. In that, there is a particular example in chromosome 5 where they connected that those centrometic regions in repulsion phase linkage connected with yield. And again, to show how recombination is not occurring evenly across all these regions, and there could be situations where very important genes are concentrated in regions of low recombination. A more recent piece of work, a beautiful work done by this group in China. They have done a complete telomere to telomere assembly of the maize genome. And among a number of very interesting discoveries, they have found that when they look specifically at the centromeric region of Missouri 17, which is a very important reference line of maize, they found 82 genes that were concentrated in these centromeric regions. And these genes, a large number of them were found to be important genes. So these are not just genes that are sitting there doing nothing for the plant. They are important, and they are controlling economically important traits. So again, to the point that we have this historical variation, and some of the variation, even though theoretically we have the opportunity for a large number of recombinations and mutations in each generation, a lot of these important economically important loci are in regions where there is low recombination, or they are in repulsion phase linkage. The other, when I talk to these introductory plant breeding classes, the other argument is like, well, there are other sources of variation that we also need to consider in a significant amount of work, especially in some of these larger genome species, looking at what is the role of the pangenome and what is the role of what we call dispensable genome. So for example, we know that in maize there are about 32,000 core genes, and but there's also has also been found that there is more than 70,000 called dispensable genes, and those are considered right, could be a real reasonable source of variation. And we know that the functional gene conservation very frequently extends across across many kingdoms. So when we look at the potential role of dispensable genes, and this is work done by Candy Hirsch group at University of Minnesota, the summary of this is that what I'm showing you here is the size of genes, and what her group was comparing is on the left side, the blue color portion of the figure is comparing all genes, so all genes in the genome of B73 compared to those that have presence-absence variation, again, as an example of dispensable genes. And then the same example for a different inbred line, in this case, PH207. And this is a very large study, including a large number of genes. And the finding in here is that genotypic-specific genes are slightly smaller, and they are generally less complex than genes that are present in both genomes. So indicating that even though there is a very large number of disposable genes that are available and potentially a good source of variation, those genes don't turn to be as sophisticated as those that are as the main genes. Now, looking at the expression of these dispensable genes, this is actually a collection of studies involving our group and others, looking at RNA sequencing, an atlas including about 80 different tissues and time points from germination to senescence. You're welcome to look at these papers, but just to show that it's a very, very exhaustive set of tissues in developmental stages. These experiments were done using solid replication and always comparing it to the reference inbred line B73. And the punchline of all of these studies is that no matter whether we are looking at B73 specific genes or those that are that have genes that are present across a collection of the core 32 lines that were evaluated in this study, the genes that are specific to an individual, the dispensable genes, whether we are talking about, again, the specific B73 or in the collection of these 32 lines that we're evaluating, those that have presence as absence variation, there is a significantly smaller or lower expression of those dispensable genes compared to core genes. Again, just showing the point that genes they tend to be less complex, they are smaller, they are not as widely expressed, or they are not as significantly expressed. And then when they are expressed, they are actually expressed in specific tissues. So this figure is also a summary from those papers in showing that when we compare the genes that are expressed across all 32 of these inbreds that were included in this atlas compared to those that had the dispensable genome. So the graph on the left, the red figure shows genes that are present in all the lines, and you see that the majority of these genes are expressed on a very large number of tissues compared to the dispensable set of genes where the majority of them are expressed in one or very small number of tissues. This type of work was also confirmed by, again, beautiful work done by Matt Huffer at Iowa State University, where they look at 26 diverse genes, doing a very detailed evaluation of de novo assemblies of these large collections of genes across these 26 diverse maize. And what they found is that private genes, dispensable genes, are expressed at a much smaller number of tissues than core genes. So some dispensable genes are useful, no question about it, but many are not functional, or they are likely to be lost in the process of breeding because of their nature in their expression. Okay, so then you might argue, well, we have a number of new resources, new technologies that can help us find those rare alleles in help us identify where variation that could be extensive outside of what is present in these in the historical variation of these breeding populations. Such one of those methods that of course has been incredibly important for the breeding community within the last 20 years or so is this idea of genomic selection. And without going into the details of the mechanics of these, think about genomic selection as being a technology where we have certain populations that are considered what we call model or training populations, where we have genotypic information and phenotypic information. And the idea is that we use that information, genotypic information and genotypic information together, to then be able to incorporate that information into models that allows us to make selections for individuals and populations for which we don't have phenotypic information, considering that the phenotype is the most expensive part of this process. So markers would allow us to make more rapid selections and identify regions of the genome where variation can be explored that, when we are doing traditional phenotypic variation, we might miss. And there have been a lot of studies that describe the utility, of course, of this technology. And some of the major highlights is that for these types of technologies, there are some features of these populations that are very important. For example, the size of the reference data set, the larger the reference data set, the better is our ability to predict the outcome of the populations that we are trying to select. Of course, the heritability of the trait, the higher the heritability, the much more progress we can make. And then what are the number of loci that affect the trait? Traits that are more simply inherited are, of course, easier to select than those that are more complex. Another important feature of genomic selection that has been universally, I think at this point, accepted is that the degree of genetic relationship between the training and the testing set have a lot to do with our ability to make progress from genomic selection. What I mean by that, this is work that was published in 2010. I just like the idea that we have all of these different traits that are shown at the same time. And what I'm showing you here are 4 different traits that animal breeders are selecting for, and they use 3 different genomic prediction models, so utilizing different statistical tools to try to optimize our ability to select populations. And what you're seeing in here, the x-axis is the additive or the genetic relationship between these training sets and these testing sets. And the theme in here is that the the prediction ability of all these populations will decrease as these training and testing sets become more distinct from each other. So the point that I'm trying to make here is that it seems like, yes, indeed, these technologies could help us identify the variability that will be hard to see, in just based on the phenotype. But these findings also showed us is that if the training sets and the testing sets are very different from each other, some of that diversity might get lost because we are not able to take advantage of the relationship between those populations in order to make progress. And in a beautiful work done by one of my grad students, we look at this population that included, what I'm showing you here is the schematic of a population. And just to say, we took 4, so the lines on the columns here are 4 pretty standard, commercially relevant maize imber lines. And then the lines that are highlighted with the red square are 4 imber lines that represents white diversity. They are lines from the Germplasm Enhancement of Maize program, which is a very important program here in the United States to bring variation, tap variation from exotic sources. So we created this factorial population that the crosses of these A lines and derive double haploids from those and then test them by LH244. So the idea of this population was we want to tap into these diverse sources of variation to bring variability into our populations that is not currently present in commercially relevant maize populations. And this is work that was done by Alden Perkins. There is a lot to be seen in here, but the point that I'm trying to make in this with this research is what he tried to do is to compare 3 different selection methods. You can imagine the red is just your typical genomic selection where all markers in these training and testing sets are considered for the selection. Then you have a situation where the blue is only using subset of the populations that are similar to each other and then selecting within those. So this is kind of the opposite of what we're trying to do, right? When we bring diversity from other populations. And then the green is a sparse selection index. And in here, what we are saying is let's just use the most useful subsets for each individual and try to optimize prediction that way. And I'm going to focus here on yield just to make it easier for you to see. And when you see the trend here is that the green, which is the sparse selection index, is always has the highest prediction ability, indicating that even though we have all of these genetic variation in these populations, in order to the greatest progress occurs when we sort of exclude that unique variation from our models, because that's what's going to make us make the greatest progress in terms of prediction. So the tools are there. The point that I'm trying to make here is the tools are there, but these tools tend to narrow us even more into what is the variability that we have accessible in this breeding population. So I call it here an improved version of the lottery. It's not really bringing us new sources of variation, but just driving us faster towards that towards that historical variation. So is infinitesimal, I started by saying is the infinitesimal model a cause or a consequence? And my point in here is that it is a consequence. Genetic variation is required for breeding success. Most existing variation that we have currently available is not beneficial in any context. There are good and bad variants that have been filtered by the process of breeding and evolution. And most of the recent variation is more likely either neutral or negative. Of course, we can have a whole series of seminars about specific situations where the context will make some of these variation be positive, but on average, neutral to negative. Minimal new variation is arising in breeding pools. And so this is sort of the conversation that I mentioned to you with my colleague Sean Kepler, and sort of the daunting moment to realize that the infinitesimal model of quantitative genetic is not a result of biological constraint, but very much the result of how we have gone and continue to go about selecting for these populations. And again, the clock is ticking. So where is this useful variation going to come from in order to help us address all of these real issues that are here and are so imminent? So my argument here is that we need to consider new pools of variation to maximize breeding success. Think about how to create large effect alleles from alleles that are identified in different ways, create maybe desirable intermediate effects, consider creative ways to diversify gene function, and how we can use cross-species transfer of gene function. Overall, just create targeted positive variation that will become a new substrate for selection. In that, including altering recombination frequencies and all of these different things that we talked about to unlock variation. So I'm going to go now through a few examples of things that have been done in this context and taking advantage of the information that is available. So this is work that came out of Jonathan Lynch's group, Penn State University. He has been working for decades on improving corn in different species, and his theme is steep, cheap, and deep. That the ideal type of roots to access useful nitrogen and water are roots that are going to grow deep. They are going to have narrower root angles, and that overall will be cheap in the sense that will have aerial aspects of the root that will make it more faster growth. So he has worked for many decades on all these different traits that are described here, and in a recent collaboration, is specifically focusing on root angle. His group, in collaboration with our group, has identified this one SNP in chromosome 1 that, when was identified, it changed root angle by 2.6 degrees. As you can see, it barely made it through the threshold of this GWAS study, but through the process of reverse genetics, so this particular SNP was associated with an important gene, CBL interacting serine threonine protein kinase 15. And this gene, through reverse study, was found to change, be able to change the angle of these roots by 10 degrees in the right background. When tested under suboptimal nitrogen availability, this gene, this mutant, provided 18% greater shoot biomass and 29% greater shoot biomass compared to the wild type, indicating, just kind of an example where a gene that was identified in a mapping population with a relatively small effect, when introduced into a different background, could provide a change that was substantial in terms of the impact both in shoot biomass and therefore nitrogen uptake. Thinking about intermediate effects, for example, this is a example of a work done in our lab by Vai, Sani, Lore, Anson Su, and Jose Varela from our group. I live in the state of Wisconsin. Wisconsin, within the United States, it's known as the dairy land. We have a lot of dairy production in the state. And the fact that we are where we are geographically, we don't have access to forages. These animals, these ruminant animals, don't have access to forage year round. So the production of corn silage is very, very important because it's a main source of energy for these animals, energy and fiber for these animals during the winter. So silage in the United States is the second largest area after corn grain. So the greatest use. And because the silage is fed to animals, we are concerned not only about productivity, but also quality. And so for many, many decades, breeders have been working really hard on improving the digestibility of different corn varieties to improve animal nutrition. And so one of those important genes is the brown midrib gene, which is a gene that affects the lignin pathway and makes corn plants that have this gene more digestible. So you can see here in the picture, it's called brown midrib because it literally creates a brown midrib on the plants that have this particular gene. So even though these genes increase digestibility of these hybrids significantly, it comes with serious issues related to standability and overall agronomics. So these plants are more digestible, but they are also more susceptible to lodging and diseases. And so for many years, breeders have been working on improving the background of these materials to make digestible corn plants. So we improving the quality of these plants that will be fed to ruminants while still maintaining the productivity and the agronomic consistency. And other examples of such modifications have been shown in alfalfa, also high digestibility alfalfa. The way the breeders have been able to create varieties that are productive, agronomically stable, is by creating RNA alleles that have optimum agronomics in elite varieties by identifying intermediate alleles. So our goal with this project was to do something similar in maize. And we did that by modifying this brown midrib. And I'm not going to go into the details of this slide, but what I'm showing you here are 5 different mutation types by 5 different events, and that were classified in what we call strong alleles and weak alleles. So weak alleles will be these intermediate alleles that hopefully will improve the digestibility of these populations while not affecting agronomics. So I'm gonna show two examples here. And again, I'm going to simplify the names of them, but what I want to show is the example of situations where you can modify these alleles to be able to have intermediate expression of certain alleles that allows you to, for example, in this particular case, improve digestibility while maintaining agronomic entirety. So here I'm showing the top is a non-transgenic version, what I call a weak allele, and then a strong allele with a much darker reddish coloration in the midrib. So the way we measure the strength of these stalks or the ability to stand was using a very sophisticated pressure piece of equipment in the engineering department that is basically measuring, as you can see in the picture, the ability of these stalks to bend without breaking. And what I'm showing you here is an example of the first two columns on the left are just W64A, which is an old inbred line from Wisconsin, just as a control. You have the wild type on the green and the brown midrib one on the blue. And what I show in the right are examples of a strong allele, the next green box in here, compared to the weak mutant. So we are able to identify these two mutants are equivalent in terms of digestibility, but the intermediate allele, the weak mutant, has a much stronger stalk, being able to allows us to do again achieve both objectives. So this is variation that is created to editing in a subtract for empirical analysis. The testing is still ongoing to identify lines that have good agronomic and improving digestibility by showing that it is possible, and having alleles in an inbred background that doesn't have this linkage drag definitely improve phenotypic, the ability to do this phenotypic analysis. And like this, you can think about many other genes. We think about flowering time in corn all the time, and many core genes are ubiquitously expressed. For example, flowering, plants that tend to be earlier, they also tend to be smaller. So you can think about what we call the early flowering syndrome, where, for example, I'm showing here the example of a plant that was modified by a single gene, this MAD S69, which, yes, indeed, is a much earlier line, but it also presents smaller root types, thinner stalks, and overall a small productivity. So how can we modify these genes by specifically improve the traits that we are interested in without affecting the expression of other genes that are affected by this QTL? So can we diversify gene function to, for example, in this case, get early vigor in late flowering plants or early flowering hybrids with late flowering yield? And so the idea that we can specifically target genes in the pathway. So just to finish up, I know my time is almost up, and so I also want to say that this is not a pitch to convince anybody that we need to specifically forget about what the sources of variability are, but to think creatively about what are our sources of variation and how we can exploit them in ways that would allow us to create opportunities for breeding to tap into and improve these traits that are so important to all of us. I will finish up by saying that this is a piece of work that was published also recently by a group of us. And when we are thinking about how to scale up these different variations that we identify in the genome, that it is important that they are done in ways that we validate the benefits of these genes in ways that are meaningful. And so the bottom line of this piece of work was to think about, there have been a very large number of studies that have investigated the impact of modifying a gene or a few genes on the yield of crops. And many of those publications are using conditions that are not conducive or reflective of what these genes are going to be doing in the field where we are interested in seeing it. So Dr. Keifel-Bart, she very nicely summarized, or she gave us what are considered to be 5 important messages when thinking about how to test these variants in conditions that are relevant to breeding. And the 5 messages that she shared with us is that studies should use standard definitions of yields. The trial should be replicated across plots, geographical locations, and years. That varieties, planting densities, and other conditions should closely match those that are found in farms. That, of course, we should always use appropriate controls, and that research should prioritize genes that plant breeders might have missed. There is a lot of studies that look at genes that are interesting but not necessarily relevant for all these challenges that we mentioned before that need to be addressed. So it is important to, in consultation with breeders, to make sure that we are prioritizing those genes that are important to us. And of course, there are several examples of efforts that are working really hard to make that happen. Many such examples in the CGIAR in variety trials across many universities and institutions around the world. I'm just mentioning the example of one that I am most familiar with, which is the Genomes to Fields initiative. And I'm pointing out here, so up to today, we have collected data across more than 280 locations, large number of experiments and plots. And this data is made available, publicly available, genotypic, phenotypic, and weather data. And the beauty of this effort, for example, is that it provides a framework that allows for testing of genes and variants in the conditions that are relevant for the types of efforts that are important for breeding. And so, for example, in the Genomes to Fields, we do that by using protocols that are standard for phenotypic data, replicating not only in terms of environments, but also field replications, using varieties, planting densities, and conditions that are meaningful for the germplasms that are being evaluated, always consistently using checks, and prioritizing how we select and what those where these varieties are placed. We know by recent studies that about 60% of the variability in the environmental conditions are year-to-year variations. So we need to understand what is it that we need to select and where those varieties are going to be placed. So I know I am running out of time, so I'm going to just skip to my last slides, or just to think about, or the main message that I want to give is that breeding has been no doubtly extremely successful. This is just one example. Again, this is corn, which is what I'm most familiar with, and the significant increase in yield that has occurred in corn in the United States for the last 100 years. So I'm not arguing in any way, shape, or form that breeding hasn't been successful. It definitely has been. My argument here is that we are at a crunch time in terms of how much we need to accomplish in the amount that it is available. So we really need to focus on where is the variation that we need, how are we going to tap into that variation, and consider the timeline and how where do we imagine that variation to be present. So when we think about the expression of certain genes, there are what we consider primarily constitutive traits, those that are expressed across all environments and all conditions. In those, I wouldn't say they are easy, but they are more straightforward to measure. Flowering time, root angle, leaf and angle shape. Now, a lot of these traits that are going to become important when we think about how do we continue to improve plant varieties across these diverse environments are traits that are going to be present only in specific conditions. For example, stomatal response, or how abortion occurs of ovules. And then things like signal transduction cascades, the example that I quickly showed about flowering. So when we are thinking about what are the traits that we need to focus on moving forward, that kind of variability is very specific of the conditions that we are measuring. And so we need to realize that those conditions need to be present, and we need to target the type of variation or the sources of variation that will improve or have the highest likelihood of allowing us to identify it. So if we're breeding for stable, broadly adapted cultivars, yes, the constitutive traits will be much more important over responsive traits. But when we are thinking about this diversity of temporal and spatial environmental stresses, the probability of identifying those traits or the correct environments or the proper environments is much more infrequent. So we need to keep that in mind, that is just going at it without a strategic thinking of what is the type of variation and in what situations it will be expressed, will reduce the efficiency of these breeding processes. So I'm gonna on my last slide, as I mentioned, there are a large number of people that I should be acknowledging, and I won't try to. This is just a picture of our group from last summer. I am completely indebted to them and the efforts that they have done over the years, and then the funding sources that allows our work to happen. So with that, I am going to stop, and happy to answer any questions.
Thank you so much, Natalia. Thank you for an excellent presentation. Please post your questions in the chat. We have time for at least 3 or 4 questions. I'm going to start with a question from Ethiopia, from Hiwan. And very simple question. It says, I get confused. Is variation good or bad? Can you elaborate this concept?
I love that question.
It's very nice.
You know what? It's not a basic question. It's one that we should all be thinking about all the time. So we need variation. We can make progress in breeding without variation, but we need to remember that not all variation is useful. I would say is if to create variation, you know, our fields here in Wisconsin, my fields, my research fields, are full of variation. That means we have a lot of crappy things. I can't make great progress if all I have in a field is a bunch, you know, 50% of my space is taken up by crappy things. So variation is very, very important. And we spend a lot of time thinking about how to maintain that variation. My point here is that we need to be extremely strategic about what kind of variation is the variation that we really need, and think about what are the most likely sources of that variation. So that is not a simple question. It's a very complicated one, and one that I try to address with this presentation, but it's not one that I necessarily have all the answers for. Not as simple. Yeah, thank you.
No, it's not a simple one. Valerio, it's not that I forgot your question, but I thought that question was simple and quite fundamental. Valerio is asking about your thoughts or opinion when breeding programs can obtain broad sense heritabilities that are pretty high, then are above the best predictive accuracy possible. So does genomic selection still make sense for those situations?
Yeah, I mean, it might make sense if the traits are still very difficult to measure. I mean, everything boils down to what is the unit that you're using to measure that efficiency. So, you know, traits can be very highly heritable in certain conditions. And I think that's the point of my last slide. In certain conditions, you might have that high heritability, but it's not necessarily the conditions where that trait will have the greatest impact. So I think in those situations is where genomic prediction, no matter, you know, we can become, you know, we can have long discussions about the best methods and, you know, the best experimental conditions. But the bottom line is that genomic prediction can help accelerate things. But a very important component of genomic prediction is that it allows to measure traits that are not necessarily accessible at all times under all environments.
Okay. Vishal here, Vishal Singh is asking, he's curious to learn how CRISPR can be integrated to further generate gene-specific variability.
Yeah, I mean, I think that is the future. I don't believe that CRISPR is the answer. I don't believe that, I mean, it's not just a matter of believing. I mean, these traits are all very complex. I walk around cornfields all the time. a corn plant, same genotype, never looks the same from this plot to the other plot. The interactions, gene to gene, gene to environment, it's not it's not at all, like a switch. But my argument in this presentation is that we can modulate big players, for example, to allow us to untap variation that otherwise will not be accessible. And that's, for example, the example of the root example, right? We identify a gene that when we did the GWAS, it barely passed the threshold. It didn't seem like a very promising one. But when you put that gene, once you identify it, you put that gene in the right tissue or with the right promoter, and all of a sudden it opens up an opportunity for many other genes to express themselves that were not able to be expressed because the conditions in that genome were not available to allows us to, get those narrow angle roots. And so that's kind of the point that I'm trying to say. We have technologies nowadays that allows us to strategically make modifications that are not necessarily the ultimate place where we want to be, but it opens up opportunities for the expression or the interaction of a series of other genes, which, again, to the first question, we just don't want any kind of variation. We want a useful variation.
Well, okay. I'm going to entertain one more question. And Matthew Blair here, Matthew is asking about genotyping costs are going down, not as much as he would like to, but he's also interested to see how you are reducing phenotyping costs, and he's interested in your machine for crushing stocks. So Matthew, please write to Natalia, okay?
Yeah, happy to talk more about it's one of those things, one of my students had a really good friend in the engineering. That machine is used for a very different purpose in the engineering department. They just happened to talk over beers one day and they said, well, what about if we put corn stalks into it? So I'm happy to talk more about that. Definitely, I mean, I don't think, again, I don't want to preach to the choir. This group knows that phenotyping is not only the most expensive, but it's also the hardest and the one that has the biggest impact in our ability to make progress. Not only, phenotyping, but how do we, again, phenotype under the right conditions and measuring the right things. And so phenotyping is very important, working very hard to making phenotyping tools more accessible. I would also say that the most important thing, or one of the important things that we are trying to do, and again, an effort with the Genomes to Fields initiative, is to standardize some of these phenotyping efforts. We could be taking much greater advantage of the data that is collected all around the world for these important traits across important sources of variation. And we simply can, because when we say, plant height, there might be 10 people sitting in a room and there will be 10 different ways of measuring plant height. And so one of the things that we are also in the work that Merit has been trying to do with that last publication I mentioned is to create not standards necessarily, because standards are hard in this context, but remind people that there are certain things that can make the data more valuable, not only for their own programs, but across the universal use of the data.
Well, thank you so much, Natalia. There's other questions that we may relate to you later on after we're done with the seminar and we look at the chat. But thank you so much. I appreciate a lot, and we all appreciate your time and your presentation. Thank you, everybody, for attending today. It's been great. And for next Thursday, we have another special invited speaker, Dr. Maria Andrade from SEEP, the International Potato Center, talking about sweet potato research and innovation for sustainable and resilient food systems in Africa. Dr. Andrade was a recipient of the World Food Prize back in 2016. So I bet you will find her talk very interesting, and please ask questions because she's one of our references at the International Plant Breeding Scene. Thank you so much. And you guys have a nice weekend. Thank you, Natalia. And I'll see you next week.
Thank you for the opportunity, and just email me if you have any questions.
Great. Thank you, Natalia.
Thank you.
Bye.